Hello guys, this is Aloysius Guy in a new tutorial, and today's topic is quite interesting. It's about red blood cells, understanding their function and how they are very specialized in doing their role in the body. And please don't forget to check the time codes in the description so you can go directly to the chapter that you are interested in. So the first thing that you need to know about red blood cells, or RBCs, as I'm going to be referring to them throughout this tutorial, they are the most abundant type of cells within our blood. So in our body, we have something called the circulatory system, and that system is made up of arteries, the red, and veins, blue, okay? Within these blood vessels, as we can see here, there will be some immune cells, these big cells, with some platelets, these small cells, and there is a lot of RBCs in our blood vessels. So if we were to separate the content of our blood based on the volume, we can see that using centrifugation, we would have three different, it will be separated into three different parts, okay? The first part is the plasma, which contributes to 55% of the volume of our blood. And plasma would contain water, protein, nutrients, and some electrolytes. And there will be a very small layer here in the middle. It's called the buffy coat. It's less than 1% of the total volume of the blood. And this layer would contain the white blood cells with platelets as well. And when we look here, we can see the erythrocytes, which is the other name for red blood cells, and they contribute to 45% of the total volume of our blood. So this huge percentage of red blood cells indicates how important they are for our body and that their function is very essential for our being. Okay, so let's now dive into the main role of RBCs in our body. So as we can see here is that within our lungs, there is something called alveoli, these small sacs, okay? So once we breathe in air, the air enters these alveoli and moves here. So these alveoli are very close to blood vessels, okay? And these blood vessels will contain red blood cells. So what happens is that the blood is moving in that way. So all this blood in blue, it's representing the red blood cells coming from all the tissues in the body just to release CO2 collected from all the tissues in the body, and then takes O2, which is the oxygen that we breathed in, okay? So these red blood cells then would take the oxygen and deliver it to the heart and through blood vessels to all body tissues. So that's why red blood cells are very essential in our body, and without them, oxygen will not be delivered to the tissues in our body, and also without them, CO2 will not be taken out from the tissues and delivered to our lungs so we can exhale the CO2. So as we saw in the last slide, RBCs exist primarily in two main states. So the first state is oxygenated state, and the second state is deoxygenated state, okay? So what does these terms mean? So when we say oxygenated state, we are referring to RBCs when they take up oxygen, okay? And usually oxygen uptake, it occurs in places where there is high oxygen concentration, okay? Such as in lungs. So when we breathe in air, there will be a lot of oxygen in our lungs, and then as we showed that this oxygen will enter the alveoli and then will passes through the alveoli into the red blood cells. So what happens in deoxygenated state then? So in deoxygenated state, what happens is that the RBCs will release the oxygen that they have, and this occurs when the red blood cells passes by tissues or sites within the body that have low oxygen level. So the oxygen will move from the red blood cells out into the tissue, okay? Usually also at the same process, the deoxygenated RBCs will take up carbon dioxide and then take it back to the lungs so we can exhale it. So the question now is, why not all the cells can do that? why only red blood cells can carry oxygen and CO2 and moves to the lung. And the reason for that is for a very particular protein called the hemoglobin. So hemoglobin facilitates the uptake of CO2 and oxygen in the RBC and also facilitates their carriage. Okay, so now we need to understand how hemoglobin is actually involved in carrying oxygen within red blood cells. So as we can see here, hemoglobin is made up of 4 different subunits, okay, 4 different chains. 2 of them are called alpha globin, and the other 2 are called the beta globin chains. All these chains are made up of amino acids, so they are all proteins, okay? Also, each subunit will have something called the heme group. So heme group is a group that is embedded within the structure of each chain, okay? And each heme group will have an iron ion, okay? And these iron ions are what's responsible of binding to the oxygen molecule in a reversible way, allowing red blood cells to take up oxygen from the lungs and also releases oxygen in the tissues. So once oxygen binds to hemoglobin, as we showed, it binds to the heme group, okay? This binding will induces a conformational structure or structural change, okay? And this change will facilitate the binding of the second oxygen molecule. The more oxygen binds to the hemoglobin, it increases the affinity of hemoglobin to oxygen, and this will facilitate the binding of the other 2 oxygen molecule. So every hemoglobin protein will be able to bind to 4 oxygen molecules. This increase in affinity of hemoglobin for binding oxygen, it's called S-shaped dissociation curve or a sigmoidal dissociation curve, and this curve it is something looking like that. Okay, so to understand and interpret this curve, the first area of this curve there is when the hemoglobin find difficulty in binding to O2, and here we say there is a low O2 affinity. Okay, however, we can see the steep increase in oxygen binding to the hemoglobin, and this here indicates the binding of the first and the second molecule will facilitate the binding of the rest of the molecules. And in that area, we say that there's a high affinity or a higher affinity for O2 binding. And then at the end, we will have the saturation area where the hemoglobin will be saturated by binding to or carrying 4 oxygen molecules, and all these molecules are in fact bound to the heme group. So now, after understanding how CO2 is carried on RBCs, now we need to understand how CO2 is carried in RBCs, okay? And the carriage of carbon dioxide is a bit different process than oxygen. So let's understand how that works. So within the blood vessels, we have red blood cells, okay? And these blood vessels will be surrounded by tissues, okay? So what happens is that CO2 will be produced by the cellular metabolism within the tissues, and these CO2 then, once they are produced, they will move into the bloodstream, and there are 3 different ways of transportation of CO2, okay? So 7 to 10% of CO2 will form something called carbamino compounds. About 70% of CO2, which is the majority of carbon dioxide, will form a bicarbonate ions, and 10 to 20% will be carried in the bloodstream as dissolved gases. So each of these processes contain different steps, and we will go through them, and we will understand how CO2 can be taken from the tissue and delivered to the lungs. Okay, so we will start by covering the first 2 methods of transporting CO2, through the body. So the first one is the forming the carb amino compound, okay, and this form a 7 to 10% of contribute to this 7 to 10% of the transportation method of CO2. So what happens is that once CO2 diffuses into the bloodstream, they move through the cell membrane of red blood cells, and then they bind to the hemoglobin, okay. However, they are not like oxygen, which binds to the heme group. They bind to the amino acids group within the chains of hemoglobin, okay. However, this binding is specific to certain amino acids like lysine, okay. So it's not like binding to all amino acids. They need to be specific amino acids so the CO2 can bind to. This compound is called the carb amino compounds, okay. The second method is the dissolved gases. Okay, it's carried as a dissolved gases, and it contributes to 10 to 20% of the CO2 carriage methods. Okay, so what happens is that simply the CO2 will diffuse into the bloodstream, and then they will just stay as dissolved gases within the bloodstream until they reach the lung, and then they move through the alveoli, and then they are exhaled. Also, it's important to note that few fraction or a small fraction of oxygen molecules will also be carried in that way. Okay, the vast majority will be carried in the way that we showed, which is binding to hemoglobin within the red blood cells. However, a small fraction will be carried in this way. So the third method of carrying CO2 to the lung is through carrying it as a bicarbonate ions. So how does this process work? So as we said before, is that here we have the blood with cells and then the tissue surrounding the blood vessels. Okay, so CO2 will be produced by all the cellular metabolism and then diffuses into the bloodstream. So what happens is that CO2 will enter the red blood cell, okay, and once they are inside the red blood cell, they will interact with the water, and this interaction will be facilitated by an enzyme called carbonic anhydrase, okay. So this enzyme will result in producing carbonic acid within the red blood cell. Once carbonic acid is produced in the red blood cell, it will be dissociated into bicarbonate ions and hydrogen ions. So where do these ions go? So what happens is that the bicarbonate ions will move through an antiporter into the blood cell, into the bloodstream, okay, and they will be there. And for hydrogen ions, they will bind to the hemoglobin, and this will stay like that until they reach the lung. So in the lung, what happens is that the bicarbonate ions will re-enter the red blood cell, okay, and they will and the carbonic anhydrase enzyme will facilitate their rejoining with hydrogen, and then they will produce CO2 again, and then the CO2 will be moving out of the blood vessels into the alveoli, as we showed at the very first of this tutorial. Also, we need to remember that this way of transporting CO2 is very essential for regulating and maintaining the pH level of the bloodstream. So after understanding this very specialized role of red blood cells in carrying oxygen and carbon dioxide, so the question is how red blood cells can do that without even having a genetic material or even a nucleus. So how do the red blood cells have globin or hemoglobin if they do not have a DNA to be transcribed and then translated into that protein? So now we're going to answer that question. So the reason that red blood cells do not contain a nucleus is because of the process that produces red blood cells. This process is called erythropoiesis, as we can see here. So the bone marrow is the main place of producing blood cells, all types, okay, including red blood cells. And the reason for that is that the bone marrow will have stem cells. They are called hematopoietic stem cells, and these cells can differentiate into the different types of blood cells, including red blood cells, But because in this tutorial we are focusing on red blood cells, so we will show the development of red blood cells only. Okay, so as we can see here, there are steps of the production until we get a very mature erythrocyte at the end. So what happens is that first stem cells called hemocytoblast, okay, these stem cells are able to differentiate into different types, or actually all types of blood cells, including red blood cells, okay. And the word differentiation, it means they are changing from one type to another. So for example, these stem cells will differentiate in becoming more committed cell, and these more committed cells, they are called pro-erythroplasts. And these pro-erythroplasts, they are cells that are committed for erythroid lineages, which means it is committed to produce red blood cells at the end, okay. And then we get into the developmental pathway of producing red blood cells. So it's split into three main phases. So the first phase is the early erythroblast, okay, and this cell is characterized with high ribosome synthesis, also high hemoglobin synthesis, with a very large nucleus. Then in the late erythroblast, what happens is the beginning of phase 2, where there is an accumulation of hemoglobin within the cell, okay, and there are also going to be some decrease in the nucleus size, okay, and that's very important, because at the beginning of phase 3, as we can see here, what happens, the normal blast will eject the nucleus, okay, which is called enucleation, okay, and here where the red blood cell loses the nucleus, okay. So the reason that the red blood cell have hemoglobin and everything of or all protein that are necessary for the function of red blood cells, the reason that the red blood cell has that is because the developmental pathway of red blood cells included nucleus, but then at the beginning of phase 3, the nucleus was ejected, and that's why it was lost. This will result in producing something called the reticulocyte. Okay, so reticulocytes are released into the bloodstream, and within 1 or 2 days, they will develop into a mature erythrocyte. Okay, so the age of erythrocyte usually estimated to be 120 days. They will be circulating the bloodstream until they're gonna be until they die, and then they are removed from the bloodstream. So now you need to ask yourself, why red blood cells have this very unique developmental pathway during which the nucleus is lost? Because the vast majority of cells in the body, they do have a nucleus and they do not lose it during their development. And the reason that the red blood cells loses their nucleus is because that's very essential for their function, okay? So when you look at the shape of the red blood cells, their shape is called biconcave discoid, okay? So having a biconcave discoid, it means that, so the word, so bi mean two, okay? And concave is just that shape. So they have two concave shapes, okay? And having a nucleus would have prevented that. That's the first thing. The second word, which is discoid, it just means that when you look at the red blood cell from the top or the bottom, they have a circular or an oval shape. Okay, so how this biconcave discoid shape supports the function of the red blood cells. So the first benefit of that is increasing the surface area to volume ratio. What does that mean? So that means that the area for contact between the red blood cell and the oxygen molecules is increased when having this biconcave shape, okay? Which means that there are going to be more oxygen contact per red blood cell, okay? And this will facilitate the uptake and release of both oxygen and carbon dioxide through the red blood cell membrane. And here is a very small, a simple schematic showing how small cubes could have a big surface area to volume ratio, compared to bigger cubes like that, which their surface area to volume ratio is just 2 times bigger. Here it's just 6 times bigger. Okay, and that's very important during the uptake of CO2, allow maximizing O2 and CO2 release and uptake. The second important benefit is the flexibility and deformability of red blood cells. So not having a nucleus and also having this biconcave shape allow the red blood cells to get squeezed within very small capillaries that are found in many places within the body. So the red blood cell will have like different shapes and squeezes through these capillaries until they reaches the other side, and then they will have their normal they will regain their normal shape again. Okay, and that would have not been possible with having a nucleus, and that's why now we see that it's very important for the red blood cell to not have a nucleus, because oxygen need to be delivered to all tissues in the body, and also CO2 need to be taken from all tissues as well. That's why red blood cells needs to reach to all tissues in the body, and their shape supports that profoundly. So after a red blood cell does its function, delivers oxygen, and then circulates the body for 120 days approximately. So they get old, and then the body needs to remove them. Okay, and the removal of red blood cells primarily occurs in the spleen and the liver. Okay, so once the red blood cells get old, what happens is that there are going to be changes in their membrane integrity, okay, and also in their receptors that are found on the surface of red blood cells. Again, another change that might occur is that the red blood cell will lose its biconcave shape, and it becomes more rigid, okay, and less able to squeeze through the capillaries. Okay, and these changes makes the red blood cells recognized by macrophages. Okay, so once the macrophage interact with a red blood cell and identify it as an old non-functional blood cell, the macrophage will engulf the red blood cell and conduct phagocytosis, and then the red blood cell will be broken down. So what happens is that the hemoglobin within the red blood cell will be separated into the globin chains, and then the globin chains will be broken down into their amino acids for synthesis, for other protein synthesis, and then also the hemogroup will be broken down, okay, and the hemogroup then will be delivered to the liver. And regarding the iron, it's either stored within the macrophage or it's released into the circulatory system, and it's used again for erythropoiesis in the bone marrow, as we already described. In terms of diseases and conditions that affect red blood cells function, there's a diverse and a wide variety of diseases and conditions. However, there are 3 main conditions. The first one is anemia, which is generally a decrease in the number of red blood cells, or just a decrease in the amount of hemoglobin in the blood. Also, you have sickle cell disease, which is a genetic disorder, okay, that affects the conformation or the structure of hemoglobin, results in producing a hemoglobin in an S-shaped hemoglobin, making also the whole shape of the red blood cell become like a sickle shape, okay. And also there's thalassemia, which is another genetic disorder characterized with a reduction in the amount of hemoglobin, and this results in producing fewer and smaller RBCs. So hopefully in the future I will be doing some videos on these conditions and diseases, and I will go into more details into their conditions. And here are some questions to test your understanding of today's tutorial. And with that, we conclude our today's tutorial. I hope you find it enjoyable and you were able to understand it. Please don't forget to like, comment below, subscribe, and turn on the notification button. Also, don't forget to go to the description where there are the time codes, so you can go quickly to the chapter of your interests. Thank you for watching.